Welcome to Human Physiology 242 Lab 5. Today we'll be talking about the lymphatic system and immune response. So a bit of an overview of what we'll be chatting about today. So first, the lymphatic system, we'll talk about its structure and function. Then we'll talk about immune response, so particularly how that relates to the lymphatic system. And then we're going to talk about an activity we're going to be doing in lab this week, blood typing. So we'll be talking about precautions associated with blood typing and then actually preparing for that lab activity. That brings me to reading. So what do you need to do? Exercise 35, the lymphatic system and immune response chapter, you need to read that in your lab manual. Then also in your lab manual, exercise 29, blood, you don't need to read the entire chapter, but you absolutely need to read the precautions at the beginning of that chapter, and you need to read the activity about blood typing. So the lymphatic system. What is the lymphatic system? As compared to most of the systems in our body, the lymphatic system doesn't get a whole lot of press. It's not as glamorous a system, so to speak. But it plays a very, very important role in our body. It's critical to our function and operation. We'll talk about why as we go through the video today. In order to prepare you for this week's lab, I spent a lot of time, as you can tell, drawing a very detailed and highly anatomically accurate drawing. You can see my extremely good rendition of a heart up here. So the heart pumps blood throughout our body, so we know that blood leaves that left ventricle. The blood is pushed through the arterial side of the vascular system, enters this capillary bed, and then returns back to the heart via the venous system, or the venous side of the vascular system. Blood contains a lot of solutes. We know that, right? Red blood cells, proteins, glucose, other nutrients. That comes down in this capillary network, and we need these capillaries. This is where the oxygen moves across those capillary walls, the proteins, glucose that we need. However, those aren't the only things that move across those capillary walls. We also lose a lot of fluid. So those are all the solutes that are in the blood, but the solvent of blood, the water, the plasma that makes up most of the blood volume, leaves those capillary walls as well. So we lose a lot of fluid from those walls, about 20 liters a day. Fortunately, physics works, so via osmotic pressure, a lot of that fluid comes back in because those red blood cells, they're too big to leave the capillary walls, right? And then there's big proteins, circulating proteins that are too big to leave the wall. So we end up with a higher concentration of solutes on this end of those capillaries. So via osmotic pressure, a lot of that fluid that leaked out here on that higher pressure side comes back in via osmotic pressure. But about 20 liters leaves on this side and about 17 liters comes back in. So you can see the problem here, right? We lose about 3 liters a day of water to that interstitial spaces, 3 liters of plasma. That's a problem. So one, it's going to make us really bloated, right? Then it's going to make us really thirsty. The average adult drinks less than 2 liters of fluid a day. So you see the math here, right? We're losing 3 liters. We're only taking less than 2 liters. We got an issue. Fortunately, the lymphatic system comes in. Ta-da! So it collects that fluid in those interstitial spaces, brings it back through that lymphatic system, ultimately returns it back to the venous system so we can reestablish the volume in our vascular network. So hurrah, right? There's a couple functions of the lymphatic system. First, it transports that leak fluid back to the blood vessels. We just talked about that. Next, it protects the body. How does it do that? Well, it removes foreign material from lymph. So when I say lymph, I'm talking about the fluid in the lymphatic system. So that's the lymph. So the lymphatic system removes foreign material from that lymph fluid. It also acts as a site for lymphocyte policing of body fluids. So lymphocytes, we'll talk quite a bit about those. So those are cells, those are the good guys in the lymphatic system that police, that find the bad guys in our system and try to get rid of them. And then the lymphatic system also acts as a site for those lymphocytes, those good guys, to be multiplied. So what are the components of the lymphatic system? Well, here's a rendition of the drawing, arguably maybe a little bit better than mine, but you get the idea. So similar concept. blood comes via the arteries, capillary network, back through the venous system. Then we have the lymphatic system here. So there's lymphatic capillaries throughout the body, adjacent to those capillary spaces, throughout all our tissues, collect that interstitial fluid and bring it back via the lymphatic system through valves and nodes, ultimately entering back to the venous systems. So the components of that system are the vessels, the tissues that make it up, the nodes. We'll spend a bit of time talking about those nodes. And then there's some lymphoid organs, so the tonsils, thymus, spleen, including those. So the lymphatic system, how is the lymph transported? Well, we talked about those capillaries that are spread throughout the body. Those lymphatic capillaries pick up the fluid in those interstitial spaces, transport to those collecting lymphatic vessels up to lymphatic trunks, and we're talking sequentially larger and larger lymphatic vessels, and then ultimately into ducts in the thoracic region, so the middle of the body, so to speak. So the right lymphatic duct, you can see it up here, leaders pointing at it. The right lymphatic duct collects from the right upper extremity, the head, and the thorax. And then we have the thoracic duct, so labeled over here. Thoracic duct collects lymph from the rest of the body. So where do they return it? Well, they return it up here at the junction of these internal jugular veins and the subclavian veins. So the right lymphatic duct enters right back here, and the thoracic duct enters right over here on the left side of the body. So you can see we're really close back to the superior vena cava, so very close to entering right back into the heart. So the other thing I'd like to point out is that cisterna chyli right here. So the cisterna chyli collects lymph from a lot of the viscera in the body, but additionally it serves to receive chyle or lipids processed by the small intestines. So that chyle, those lipids are too big to get across the capillary walls. So the lymph system steps in and helps out. So that chyle is able to be picked up in the lymphatic system and be transported via the lymphatic system, and then ultimately enter that vascular network to be circulated to the body, so that chyle can get to the spots it's needed throughout the body. Yeah, so receives chyle. How are things transported in the lymphatic system? Well, the lymphatic system is similar in structure to the most similar to veins, but it has three tunics, like the vascular system. Those tunics are generally thinner walled, but they also have more valves. So like the venous system, lymphatic system is full of valves and more of them. So that fluid from the interstitial spaces enters these collecting lymphatic vessels, and that fluid is moving this way through the body, or through the lymph system, passes through the leaflets. So you can see the leaflets of the two valves. So there's a bit of an opening here, right? So the lymphatic system can come through, and then if there's any pressure on this side pushing this way, then it's going to cause those leaflets to clamp shut, so there's no way for the lymph to flow backwards. So it can only keep progressing this way. So just like we learned for the venous system, we rely on a pumping action, so the skeletal muscle pumping action, or diaphragmatic pumping action, the changes in pressure in the thorax, generate a pumping action, so it's constantly pushing that fluid. And since it can only go one way each time there's some pressure introduced, then fluid is continuously pushed through that lymphatic system. So in the lymphatic system, we have nodes. We have nodes throughout. You guys are probably familiar with nodes. So when you get sick, you can see some of your key nodes, probably in your neck region. If you go into a physician, they're gonna look for inflamed nodes or palpate those lymphatic nodes. So you're familiar with a few of them. However, there's thousands throughout the body. You just can't see most of them. They're buried in the tissue. They're along those lymph vessel paths because the fluid comes into those nodes and then exits, and we'll talk more about that. They're generally embedded within connective tissue, so that's why you can't see a lot of them. But there are a few sites where they have perfused, so where you have a lot of lymphatic nodes. So the first is that inguinal region down here, the axillary region up here, and then the cervical nodes. These are the ones you may arguably be the most familiar with. So when you're battling flu or some other virus infection, those nodes on the side of your neck get enlarged. So you'll be able to palpate those, or a physician will palpate those to evaluate your condition. That brings us to, what is a lymph node? So a lymph node, let's talk about structurally. So there's afferent lymphatic vessels. So your tissues being out here picks up that fluid from the interstitial spaces, that lymph, transports it via that lymphatic network up through all the valves, right? Skeletal muscle pump. The fluid can only move one direction, so that enters the lymph node. So these lymph nodes serve to function almost as on-ramps to a highway or an interstate. So we have a bunch of different on-ramps entering here, and then only a few leaving. So this is the highway, the interstate leaving. So these are bigger vessels. So we have a lot of small feeders coming in to bigger networks, to the highways as they leave via these efferent lymphatic vessels. And you'll notice on either side they both have valves. There's a lot more stuff that goes on in the middle here, and we'll talk about that just a little bit more in a bit. Let's step aside and we'll talk about the immune response. So the immune response of the body. Immune response refers to the ability of that immune system to recognize something that's foreign and act to destroy it. So you don't want foreign pathogens proliferating in your body. You want to be able to get rid of them, and that's what the immune response does. So it's a systemic response, so it acts throughout the body. It's not localized to one particular spot. So an immune response acts through your whole system. And its goal is to protect us from bacterial infections, viral infections, bacterial toxins, and even cancer. So there's a couple of characteristics of the immune response. So it's got a memory, it's specific, and it's self-tolerant. So what do those mean? Your lab manual used the example of chickenpox. So if you had chickenpox as a kid, your body remembers that, and you won't get chickenpox again. So it has a very, very good memory, and that memory is very specific. So it remembers that particular virus, and next time you're exposed to it, it can go seek out and destroy it before you have any reaction to it. The other really important feature of our immune response is that it's self-tolerant. So what that means is that our immune response does not attack our own cells, our own body. So we need that self-tolerance. If we don't have self-tolerance, then our immune system turns on ourselves. It attacks our own tissues, our own body, our own cells. And there are autoimmune diseases out there. You guys may be familiar with MS. That's an autoimmune disease. So self-tolerance is an important characteristic of that immune response. So for immune response, antigens. So those are molecules capable of inducing immune response. So when we have a pathogen, something foreign in our body, it presents an antigen, so some molecule on its exterior that our body can recognize as foreign. And that is what our body cues in on to induce that immune response. Then we have lymphocytes. So these are cells that recognize those antigens on those foreign invaders and get that immune response initiated. So these are part of the lymphatic system, and they have specific receptors on their surface for those antigens. So there's three types of lymphocytes. So there's natural killer cells. So these are part of our innate immunity. So these are kind of broadly acting cells that are ready to go all the time, so to speak. Then we have our adaptive immune cells. So these are T cells and B cells that target particular antigens. So as these T cells and B cells develop, they undergo maturation. So that's where they become immunocompetent. Meaning they're capable of inducing an immune response. And part of that maturation process is becoming self-tolerant, like we just talked about, where they can recognize our own bodies. So let's talk about antibodies. So these are what are on the exterior of these B cells to recognize the antigens. So they bind to a specific antigen. So they target a specific antigen on a pathogen to initiate that immune response. So they have antigen binding sites, and they're made up of a common structure. So they have 4 protein chains. So they have 2 constant regions. So the constant regions are here in the light blue and the light purple. Constant regions, then they have a variable region. So these are the regions that will change depending on the specific antigen they are going to target. That brings us back to the lymph node. So I glossed over the stuff in the middle here. So one of the things I'll point out is the trabecular structure here. So hopefully that sounds familiar to you from back when we were talking about bone. So the insides of bone are full of trabecular bone or spongy bone made up of trabeculae. So it creates these voids or spaces. Well, in lymph nodes, we have these voids and spaces, and they're full of B cells and T cells. So the lymph system transports via lymph particular pathogens that might be present in our interstitial spaces in our body. They come via that lymphatic system, into these lymph nodes, where the T cells and B cells jump all over them, destroy them. So these are the police, the good guys that get the bad guys here, hopefully before they can travel to the rest of our body. Either way, these T cells and B cells can then multiply here to facilitate that immune response. So when the sides of your neck are swollen, when you have those protruding lymph nodes, that's because we have those rapidly multiplying T cells and B cells. That brings us to our next topic, which is agglutination. So agglutination is a clumping together of particles. That's part of our immune response. So it's where our foreign invaders are clumped together via the immune response, and it causes them to fall out of solution. That clumping together, well, why does it occur? Well, it increases the efficacy of phagocytosis. So the clumped together cells means that more foreign cells can be eliminated at once. It helps us get rid of those pathogens more readily and more quickly. We can use this phenomenon of agglutination for blood typing. So red blood cells will agglutinate. So if they're attacked by antibodies, they'll agglutinate, they'll clump together, and they'll block flow. So this could be a bad thing, right? We know that capillaries really only slightly bigger than red blood cells. So if they agglutinate, if they're clumped together, then they can't progress. They can't move through a capillary network, causes a blockage of flow, and then a starvation of those tissues from oxygen and nutrients they need. That agglutination is caused by the interaction of antibodies and antigens. So what we just talked about, the antigens, the markers on those pathogens, we have antibodies on our lymphocytes that target those antigens and initiates this agglutination response as part of the bigger immune response. So we're going to do blood typing. So again, exercise 29, please read the precautions at the beginning of that exercise. We're going to be working with blood during lab. So you need to be well versed in being safe. So safety glasses, gloves, make sure you handle only your own blood. Don't reuse supplies. So there's a bunch of precautions that I want you to be familiar with before we get to lab. So we're well versed to be safe and have a good experience. So blood typing, what are we going to do during this week's lab? And what is blood typing? Blood typing uses antigens. So the same concept we just talked about on pathogens, blood typing, when you go into a doctor's office to have your blood type checked, it relies on the presence of antigens to be able to determine what type of blood you have. So we have antigens, they're present on the outside of those red blood cells, they're part of those red blood cells. Your blood, you have a particular type of red blood cell, and then within your blood, those red blood cells you have are accompanied by antibodies in the blood. Hopefully, those antibodies are self-tolerant, right? That means they aren't going to attack your own red blood cells. You don't want your antibodies in your bloodstream attacking your red blood cells, so they're self-tolerant. But they will react against other blood types. So if you get a bad transfusion of the wrong blood type, you could have a reaction where you have agglutination occur, and you get clumping together your red blood cells, blockage of capillary flow, and your tissue starts dying. That's why blood must be very carefully typed before you receive a transfusion. You must know what type of blood you have. And I'm sure most of you at some point in your life, when you've been to a doctor's office, have had your blood type checked. It's important to know that. So there's multiple different kinds of blood typing systems, different types of classification. The most common that you've probably heard of are ABO and then RH. So let's talk about an example. So if you have type B blood, what that refers to is that your red blood cells have a B antigen. It's just been named B, have a B antigen present on the outside of that red blood cell. Circulating in your bloodstream are anti-A antibodies. So you don't have type A antigen on your blood cells, so you're just fine having that antibody type A circulating throughout your blood. It's just fine. So you are self-tolerant, right? So during lab, we're going to take some blood samples. We'll put some drops of blood out, and then we'll expose them to different types of antibodies. So if we put in antibody A, that's this column, antibody B on this column onto your blood. So let's go back to the example of you have type B blood. So here's a spot of your type B blood. If we put antibody A on that blood, should be the type that you already have in your bloodstream. So just fine. So that blood is still doing just fine. If, however, we put in antibody B, which is not the type of antibody you have in your body, that antibody B recognizes that B antigen on your red blood cells, and you get this agglutination, so this clumping together of blood cells. You can see the clumps pretty easily here on this slide, and you recognize that that's not going to be so good when it enters your capillary networks. You're going to get blockages of your capillaries. All right. So the idea is that you need to know what type of blood you have and make sure that a transfusion of the wrong type is not received. We'll go through the differences of the different type of bloods or the different blood types and what antibodies they have or don't have, what they react to, etc. We'll also do a test of your RH, right? So that brings us to the end of today's video. So what did we talk about? We talked about the lymphatic system. We went through the structure and function, all those vessels and nodes and the ducts and how it re-enters the venous system and how the pumping action works, etc. Then we tied in the immune response to that lymphatic system. So we have both innate and adaptive aspects to our immune response. There's different types of cells. They rely on antibodies and antigen to initiate that immune response. And those B cells and T cells reside back in those lymph nodes throughout our body. And the lymph system feeds via lymph, feeds those pathogens to the nodes where the T cells and B cells can be exposed to those foreign invaders and initiate that immune response. So the two systems are integral to each other, or the two aspects are integral to each other. Then that brought us ultimately to blood typing. So we're talking about blood typing because this is a good way to observe antibodies at work in our blood and how that immune response is initiated. So that gives us a chance to observe that in lab. So with that, make sure you do your reading, and we'll see you in lab this week.